Next up is Colin Hill. I knew Colin when I was at Biogen. We had some interactions. I think we did actually do a collaboration with his company, but I can't remember the details. But he'll fill you in on the background and give you a really interesting talk about the use of artificial intelligence, which, as you know, is a well-used word, two well-used words which mean different things to different people. So I'm looking forward to being explained to.
Thank you. And good morning, everybody. So I'd like to thank the Larrys, so Larry, and Meredith for inviting me to speak this morning. So I'm going to talk about digital twins, and I promise I'm going to define what that means. There's not going to be any jargon here, hopefully. And how we're using these digital twins to discover and develop new drugs for rare diseases. So what problems do we have to solve to better treat and even cure rare diseases and other diseases? First, you have to figure out what's the right drug target to go after. What gene or gene product do you have to hit to reverse the disease? So that's question one. Question two is, what is the right drug against this right target? Assuming you've found and validated the place, the Achilles heel in the disease to knock down, now you have to make a drug against it. And then the third, when you have the right drug against the right target, you have to now figure out which patients are going to respond to this drug. So nothing groundbreaking. The question is, where can AI play a transformative role in solving these problems? So AI is in the news all the time. It's made significant strides in a little part of the world of drug discovery and development, and specifically that's in the area of drug design, whether small molecules or large molecules. You've probably heard about Google AlphaFold from DeepMind. You've heard about companies that have now raised even $1 billion in their initial financing. That just happened 3 weeks ago. And then just sometime last week, Google DeepMind released AlphaFold 3 that's now starting to predict interactions between proteins and other structures. So lots of progress. However, AI has really struggled to make progress in the first problem of how do you discover the right drug target? Because of course, if you're attacking the wrong drug target, even if you really well design the inhibitor, it's not going to help you. You're aiming at the wrong thing. It's also not made a lot of progress in better selecting patients for clinical trials, as evidenced by this chart here. So after all these decades of research and progress and development of data and technology, even people calling for bans on AI research for a number of months, we have not been able to budge the clinical trial success rate. 8%. So the best ideas from preclinical studies that enter clinical trials, less than 1 out of 10 times does it ever make it to patients. So how can this be? And can we do anything about it? So what we're looking at here is one of 1,000 type of posters and pictures that molecular biologists use to show the interactions between genes and gene products. This particular map is a map of cell replication in cancer. So it's very complicated, very detailed. Almost makes it look like we understand what we're doing. So just a question here. What percentage of the circuitry of human disease on a genetic and molecular level, meaning across the genome or proteome, what percentage do we think we know? Any guesses from the audience? 0.005. 0.005, wow. Okay, so that's a really small amount. Anyone else? 10%? You said half, 50%. Oh, okay, great. So we've had from 0.05 to 10% to 50%. So the answer was, and it really hasn't changed, so our best estimates of this are actually 5%. Right? So despite the fact that we have these nice looking maps of how everything works, and there are parts of biology that we do understand well, cell replication, programmed cell death or apoptosis, lots of other things. So after tens of thousands of papers and 80 years of research, 95% is missing. Well, it's not missing, it's just hidden, like it's there, just God didn't give us the blueprint in case we got sick. So we've had to make these discoveries one by one by one. Each link is likely someone's PhD dissertation. So if we're missing, if 95% is hidden to us, no wonder we have such a hard time in discovering and developing new drugs for these diseases. It's amazing anything's worked today. So the question is, what can we do about it? So it starts with data, and even data specifically from humans. So it took 11 years to sequence the first genome. Back in 2001 is when it was finished, at a cost of $3 billion. Now those costs have come down to on the order of a few $100s and a few hours. And correspondingly, efforts to generate high throughput molecular profiling data at the mRNA level, the proteomic level, have also continued to advance in major, major ways. And these are a number of the large-scale multi-omic, meaning multiple layers of omic information, DNA sequence variation, gene expression, proteomics, specific to various diseases, and some of these are major government efforts. You're going to hear later today about the All of Us program. So it starts with data, huge amounts of data that we feel crossed the tipping point only about 3 or 4 years ago, where there's now a large enough sets of integrated data, multiple layers from the same patients in a given disease. The second thing that's been going on quietly is many decades of Moore's law, producing ever faster computing power for less and less money. This has been going on so long we almost take it for granted, but something special happened over the last 5 years. The computing power reached a level above and beyond what you could do, what you needed for AlphaFold, beyond what you needed for ChatGPT-3, reached a level that allowed us and others to start to reverse engineer digital twins, which I will explain what they are, straight from human data. So roughly 3,000 exaflops, that's a lot of computing power. And then in the world of AI, while deep learning and large language models dominate much of the AI discussions, certainly dominates Silicon Valley, there's another type of AI that's been developing for a number of years and decades that is really the bigger deal for our world and for the problems we're trying to solve. And that's causal AI. It's led to a couple of Nobel Prizes over the last number of years. The Nobel Prize in Economics 3 years ago. Some years before that, the Nobel Prize in Computer Science, the Turing Prize, went to Judea Pearl. For now a formulation, a methodology that could allow you to start to infer cause-effect relationships straight from observational data. For many decades, people didn't think this was possible. It wasn't even a good question. Turns out you can do this probabilistically. And so we've been using this framework, and we took 2 decades to develop a technology platform based on it that now allows us to start to discover cause-effect relationships straight from data. So it allows us to test in parallel a vast number of hypotheses of the form, does A cause B, or does B cause A? Or does A and C together drive E and F? So it goes beyond just answering the question of what happens when I see a certain clinical outcome, what do I see of the molecular levels? This is now trying to answer a do question. Going back to some of the discussion yesterday around diagnostics and large-scale cheap proteomics for healthcare, for diagnostics, you don't necessarily need causality. You just need lots and lots of data to find those patterns. When we're talking about the world of intervention, we need causality. We need to know what is causing and driving the clinical outcomes. That's why we work so hard to now try to do this causal inference. Now, this kind of process is easy, trivial, at the level of 3 or 4 variables. You can do it in your head if you were given output data and you needed to infer how things were connected. Problem is, when you get to now thousands of variables, you end up with an astronomically large number of possible hypotheses. This is where you need technology to solve it. And so we have developed and scaled a platform that's now capable of doing this reverse engineering of the known 5% of the circuitry and the hidden 95% directly from large-scale multi-omic data. And that's what we call the digital twins, or specifically Gemini digital twins. It's a specific definition. And these Gemini digital twins are computational representations of disease that capture a critical mass of the genetic and molecular interactions, the known ones and the unknown ones that are causally driving clinical outcomes. Starts with multi-omic data, like what you see on the left. This is specifically from the COMPASS study from the Multiple Myeloma Research Foundation, whole genome sequencing, RNA-seq, miRNA-seq, immunoglobulin profiles, cytogenetics, and clinical outcomes. That data is normalized, put into a data frame of patients across the molecular variables and clinical outcomes. And then the data is entered into our machine to enumerate all of the building blocks. That's the first step, is now scoring all the 2-way, 3-way, and 4-way interactions that are possible given the data, and scoring those with the p-value. And those interactions are scored based on particular mathematical forms. So at the top, maybe I'll try to, well, I think it's a little bit hard to draw. At the top of that box where A and alpha are driving C with the particular mathematical form, that gets scored with a p-value, which means what's the probability of that interaction given the data. We do this for trillions of these building blocks. Then the machine reaches into this basket with the trillions of jigsaw puzzle pieces to attempt to assemble the whole network model. Each hypothesis is then scored against its ability to generate and reproduce the data on the left. Depending on how well it does, it gets penalized. Parts of that hypothetical network are ripped out, new parts are put in, then it's scored again and again and again. This is farmed out over thousands of processors. So it scores a huge number of these network hypotheses to end up with a stable set of models that is the best representation of the system that would have given rise to the data. So these Gemini digital twins are now filling in the hidden 95% of circuitry. Here's a schematic. At the top, the light blue triangles are the DNA sequence variations. The black arrows are the known 5% of interactions that we end up reconstructing as a positive control. The red arrows are the newly discovered interactions that have not been discovered yet in the wet lab. They don't exist in the literature yet. And they're not going to exist in the literature in the normal way for another few hundred years. But we're now discovering that straight from the data. The blue circles are the mRNA and proteomic variables, and the purple diamonds are the clinical outcomes. So we've now reconstructed these systems computationally, and because they're constructed straight from human data, they're becoming a better and better proxy than animal models or cell lines or stem cells. And it allows us to do something remarkable now. It allows us now to perform virtual experiments based on real human biology, where, for example, we're going to run virtual siRNA knockdowns, knocking down each and every mRNA in the system to discover which ones are driving one or more clinical outcomes. So you see on the top left, We're pointing to a particular mRNA that's on the path to the clinical outcomes. When we knock it down, it's now moving from this is a proxy for tumor size, so cell count, from a value of 13, that's the red histogram, to the blue at a level of 6. So that's telling us, knocking down that particular gene product appears to be a good drug target. It's reducing the size of the tumor, and we're discovering that straight from computation. Versus this other component on the right, which might be moving a lot on a somologic scan or on a gene chip, but notice it's not on the critical path to the clinical endpoint. So causation versus correlation, and lo and behold, when we simulate the knockdown, it does not move the size of the tumor whatsoever. So by this methodology, we're discovering needles in the haystack, and we're doing it in a system that's becoming a much more accurate proxy for human disease biology than animal models ever will be, especially for certain diseases. We're also using this to discover these novel drug targets orders of magnitude faster and cheaper than what you can do in the wet lab. And of course, we cross-validate it in digital twins made from different patient populations. So then the big question is, well, does this work? It sounds awesome, but does it work? So one quick example before we talk about how we've applied this to discovering and developing a new drug for a rare disease. So here's a rare disease, somewhat rare disease, called multiple myeloma. The largest patient registry in the world in multiple myeloma was created a few years ago. 1,100 patients, 90 sites worldwide, and the COMPASS study has these multiple layers of data. I just described them in the previous slide. And this is more than a typical type of study. It was probably one of the most complex data sets in cancer for a long time. This data was run through our system to now reconstruct a digital twin. And what I'm showing you on the right is just a particular subpopulation and a particular subnetwork of the digital twin. And it contains one of the key groups, the T1114 mutation patients. The orange are the RNA-seq variables. The red are lab measurements. The yellow are demographics. And then you see in green stem cell transplant. It's still part of standard of care for multiple myeloma patients given in the first line. And then you see the clinical outcome at the bottom. So what we did in this digital twin is we simulated against these patients stem cell transplant on, stem cell transplant off, to see, can we discover a marker that's going to tell us who's going to respond and not respond? These are very invasive treatments. They're very expensive at $500,000 a piece, and the side effects are really bad. So we did this, and we discovered that a particular gene product, a particular mRNA, CHEK1, that when it was below a level of 10.3, it was conferring 20 months of survival benefit, versus when that expression level was above 10.3, it was giving no benefit. You see the overlapping error bars of those two curves. So we thought this was rather interesting. There was a clinical trial being run at Dana-Farber in Boston where patients were being randomized for stem cell transplant. We showed them our result. They agreed to unblind the data. Lo and behold, it validated that result of 20 months of survival benefit based on the level of this biomarker, and zero benefit when it's above. Okay? So just a quick example of, to answer the question, well, does it work? Do you have examples? And yes, lots of published papers. This was presented and published at the ASH symposium, the hematology symposium a few years ago. So what does this mean for rare diseases? With single gene causes, this should be straightforward, right? So here's a couple of examples of rare diseases. Sickle cell disease, which you heard about at the prior talk. A single mutation is enough to drive the disease. Huntington's, a single gene driving the disease. And so the question is, while the cause of these diseases with these single gene mutations seems relatively straightforward, why don't we have effective treatments for them all? It's been elusive. So the cause is not always the cure. Now, several months ago, there was a gene therapy that got approved from CRISPR and Vertex for sickle cell anemia. It's great, great advancement. But those are still the exceptions to the rule, even in these diseases. And it's because, at the end of the day, there's still a complex network of interactions driving how these diseases are driven. Larry. Okay. So let's focus on Huntington's for a few minutes. So the first Huntington patient was discovered in 1872 by George Huntington. Then in 1983, Jim Gisela and the team at Mass General Hospital mapped the Huntington gene to chromosome 4. So it's the first disease-associated gene to be mapped to a chromosome. Really big deal. Then a team also at MGH, Marcy McDonald and team, then identified there's a particular repeat, a CAG repeat in the Huntington gene that's ultimately driving the disease. It was a really big deal. So it was identified as IT15. And they showed that IT15 is being transcribed into mRNA and turning into a mutant protein. And the other thing they discovered, which was fascinating, is that there's a critical number of these repeats that determines whether you have the disease or not. So it turns out that if you have less than 35 of these repeats, you're fine. And lots of us have CAG repeats in our Huntington gene. It's not a problem. For those who have 40 or more, they have the disease. They're going to get the disease. They're going to die from it. But they're not going to die from it at the level that they're born with. What ends up happening is that above 40, it's unstable, and 40 turns into 74, turns into 87, eventually crosses 150, and that's when the motor progression and the cognitive decline really begin. Okay. So the question is, with all of the understanding we have now of this gene, how it expands the CAG repeat to become pathological, why don't we have an effective drug for it? So the question is, can we use these Gemini digital twins, the multi-omic data and causal AI, to discover a much more effective treatment for this so-called simple disease? So where do we start? We start with data that the Cure Huntington's Disease Initiative has sponsored. TRACK-HD and TRACK-ON, these were the largest studies of their kind, straight from patients, mainly from blood. Then Enroll-HD was even larger, and there was actually a subset of this where very deep molecular profiling, including proteomics from SomaLogic, was used, along with biomarkers such as NFL and others. So our team took the TRACK-HD data, integrated that data into a patient data frame, ran the data through the machine, the REFS platform, the causal AI platform, to generate the building blocks, to now reverse engineer the networks, and to end up with now an accurate Gemini digital twin from the data that's now filling in all of the hidden circuitry as well as reconstructing some of the known circuitry. This is what the specific digital twin looks like. I know it's hard to read. The thing to take away from the annotations is that this has roughly 23,000 nodes, roughly 5.3 million interactions and edges. So it's super complex, well beyond even what a human brain can make sense of. And notice in the graph near the bottom, you have the two clinical endpoints, TMS, which is a measure of motor function decline, and SDMT, which is an integrated measure of cognitive decline. And you see various demographic variables. So now the question is, can we use this digital twin? Can we simulate interventions into this digital twin to discover hidden drivers of disease progression? And that's exactly what we did. And so this is just showing a schematic of knocking down each and every RNA, measuring its impact on, in this case, the motor function. We then took the top 10 of these targets and going through a process of Mendelian randomization, analysis for other biological relevance, and druggability, we ended up prioritizing a particular target and discovering a small molecule drug that's able to inhibit this target. And so what you see on the left is how this particular target, through a network of interactions, some of which were known, many of which were unknown, is driving motor function. On the right, that same target, going through key biomarkers such as NFL and also other variables such as caudate volume of the brain, are driving cognitive decline. So here, in a completely hypothesis-free way, this was not discovered by one of our researchers having their own just hypothesis as to what might be happening. Those expert-driven hypotheses can be great, but there's way too many of them to really land at the answer. So this was completely machine-driven, data-driven, that this target was discovered and a small molecule drug against it was created. This target is overexpressed in the brain, which is great. And this graph is just showing the fact that this drug is inhibiting cell proliferation, right? The replication of DNA, which is really core to the mechanism of the drug, while at the same time not affecting the metabolism of these cells. So it's showing that it's safe on a cellular level, but it is inhibiting growth. And we are now in the middle of doing experiments to, one, validate the causal network structure. These are being done right now. Also running PK/PD studies to show it's getting into the brain at the levels we want. We are also running experiments to validate the ability of this drug to slow the CAG repeat expansion. Remember, the disease doesn't become a bad thing until it starts to become unstable, and the CAG repeat that might start with a level of 40 or more until that gets to over 150. So it's not a matter of just knocking down the Huntington gene and a gene therapy approach. Can you stop the expansion from happening? And that's what this experiment will soon show. And then the last is mouse models that will show the phenotype that we expect to see in terms of Huntington's. So these are the assays that are underway to now advance this drug towards a phase 2A clinical trial. That is the goal, right? This would be the first transformative drug, say in decades, if not ever, for this disease where the underlying gene, the cause, was discovered over 30 years ago. So is this a new approach, a new way of discovering novel treatments for rare diseases and other treatments? And that's our assertion. So to sum up what I've described here, the cause is not always the cure. Despite being defined by single gene mutations, many of these rare diseases have still evaded having an effective treatment. Advances in multiomics, supercomputing, and causal AI are starting to unravel these complex mechanisms that are unknown, that are hidden. And by creating these Gemini digital twins, we now have a chance of doing experiments in a very different way, but based on real human biology. And this is enabling us to now run these experiments at scale in silico and then compare to both wet lab experiments and then ultimately clinical trials. And so we're really excited about the progress we're making of advancing this discovery into the clinic. So what does this all mean, taking a step back? Have we reached a tipping point of human multiomic data, supercomputing, and AI that's now allowing us to start to unravel the mysteries of these diseases, not just one by one, but now to do it at scale, such that we can now start to discover and develop drugs in a fundamentally different way, and we won't have to show slides that show an 8% success rate in clinical development. That really is unbelievably bad and persistent. So why does this matter so much for various rare diseases? So my parents each had the sickle cell trait, which means they had one of those genes mutated, one element mutated. So that meant there was a 1 in 4 chance that one of their kids would get the disease. So my brother has the trait, which is what you'd expect, that he just got one of those copies. I got lucky, and I actually have nothing, not the trait. My sister got unlucky, and she actually has the disease. And so this is my sister, Bonnie Lynn, who lives in Toronto. This is on a beach in Martha's Vineyard somewhere, and that's me in sunglasses. So one of the reasons why this is so important to me is my sister. Now, she has a particular mutation that means that's been a bit protective of the disease. So she's now 47 years old and has had a relatively mild trajectory so far. But it's very important to me that we now use data and technology to find better treatments in a much more predictive and rapid way than has been possible before. Thank you.
Well, just before we go to the audience, I'm going to take the moderator's privilege and ask you a question. You did a lot of virtual knockouts. What about not knock-ins, but upregulating the expression of particular messenger RNAs and seeing what the consequences of that was? Because a lot of the mutations are gain-of-function mutations, not loss-of-function mutations.
Great question, Tim. And so the team always does both directions of the perturbations. They always do the knockdown of the mRNA by 95%, but they also do the increase in various increments of 50% and close to 100%. And we often do find sometimes some surprising impact from actually increasing the levels.
And you get as clean a data as you get with the knockouts?
Absolutely.
Okay,
yeah.
Cool. Thank you. What do we got? There's one there next to the man in the red shirt, just behind Larry. Now, you've asked one already. I wasn't looking at you. It's for the person next door. Giving some people the, you were representing them. All right, them, you can talk.
You
just asked a question already, that's why I'm giving other people a chance.
I just was wondering if you showed a plot that seemed to have more than one gene on it that could have been a candidate, and I'm wondering if you had looked at the other ones experimentally as well, or did you just choose the one?
There's actually 4 that we're really excited about in Huntington's that we are advancing. But that particular one had some very special properties in terms of the thesis by which it was slowing down the CAG repeat. And it's a good question because just so maybe people know, we were a platform company for the longest time working with many companies in the industry. We only recently made the shift to becoming a drug maker. And so the fact that we have to focus on a very small number of hypotheses is a real shift for me. It's exciting, but our ultimate goal is to be able to really bring a large enough number of drug candidates forward based on this completely data-driven and hypothesis-free approach.
I was looking to see what we can see. There's one up there in the back.
Thanks. It's really cool, actually, Colin, to see what you had proposed 2 decades ago is actually coming to fruition now. It's amazing. Congratulations. So 2 quick things. One, the refs models are derived completely free from any bias, but you use the literature to validate them. And I'm just wondering how much literature is actually wrong in its own right, and do you see those problems when you put this together?
Yeah, so we assume at least half the literature is wrong. But there's the canonical, what we like to call at ATIA, the gold-plated biology that all the experts agree on is what's there. And we really use it it after the fact. We don't use it in building the models. We don't want to be biased by it, but we do use it after the fact as a positive control of do we recover XYZ relationship. But we don't put a lot of weight on it, I guess is the real answer. It's important that we recover some of the key pieces, but once again, the vast majority of what's getting discovered and where the power comes from is from newly discovered linkages that we really look to other digital twins to validate, because they're also inhuman and so on.
And the other quick thing I just wanted to add, I think it would be interesting to look, one of the most striking things that came out of the ENCODE Consortium's publication of all their thousands of whole genomes is that there's a lot of people running around with serious lesions in genes, rare disease genes that are perfectly fine.
Yes.
And so that would be a very interesting cohort to add to this mix.
Yeah, 100%.
Larry.
So I'd like to follow up on Tim's question. I never thought about this. There's a big group in Colorado working on Down syndrome, which you've thought about. And there's a few hundred genes on chromosome 21 that are present at 1.5x, as you'd expect, at the right number for 3 copies instead of 2. And you could then, in your programs, do the digital analysis to see if you could figure out the causal gene, because nobody really knows that. I mean, the group in Denver is close to understanding it. Have you ever done that? Or?
We have not worked on Down syndrome yet. If there was a large enough set of patients with profiling, then we absolutely could rapidly make a digital twin and do that experiment.
Well, one of the one of the interesting things about the Downs is you triplicate the APP genes, so they're all very prone to Alzheimer's disease as they get older if they get to that level. So it would be a very interesting cohort to look at. And Tom said they've got all the omics data that you can look at.
How many patients, roughly?
Thousands. Well, it's big.
Okay. Oh, wow.
Other Larry.
Yeah, so I want to use that as an opportunity to ask you about, for you to illuminate us on an AI question. So these causal networks, for the most part, require binary variables in the nodes. They're either on or off. They're not they're not real numbered. You have to have a probability that it's on or off, but that's what the causal networks infer, right?
Well, actually, so we have continuous variables in our framework. So
you have to discretize them.
We don't have to discretize in our framework. In certain other approaches people do, they do have to discretize, and I used to work on those Boolean two-state things back when.
Okay, so in general, the causal approaches look to see relationships between random variables, which are largely either on or off, right?
Yep.
So I'd like you to use the Down syndrome example, where the change is a quantitative change, and it's important that it be that particular quantitative change, but not only over one variable, over a lot. So would you talk a little bit about how you can use these kind of random variables that are kind of binarized to model something like that, and what some of the challenges are?
Yeah. So great question. So the way that DNA sequence variation is represented in the models is discrete, as you'd expect. The mRNA variables, the proteomic variables, are continuous. So if there was either a hypothesis of the upregulation of these 3 gene products having a big effect, there's no reason that'd be straightforward to model and do the simulations in this framework. Now, a more interesting thing, if one was going in without having any hypothesis as to what genes, what are the factors and which combinations, you would just simulate all of the 2-way interactions, all the 3-way that you could do in terms of perturbations, and that would still be a relatively contained experiment. The key thing is there enough data to reverse engineer the digital twins? And if the answer is yes, and it sounds like the answer is probably yes, then the simulation should be straightforward. We should be able to do that.
So one more.
So I'd like to extend Larry's question in one way, and thank you for a wonderful talk. It's really inspiring. If I understand correctly, the way you're applying the causal models is you're using population data to infer the connections that you think are real in
Yes.
the digital twins.
Yeah.
I wonder if you'd speculate a little on, can you imagine or have you thought about extending that to longitudinal data so you can handle cases where the time constants in the links are included, because it's clear that a population level model of this interacts with this is inadequate to predict what happens in a single patient.
Yeah, a great question. And I didn't get into the details of some of the data. I would say about a third to half of the multi-omic patient data sets we get are longitudinal. So in Alzheimer's, for example, the ADNI data set has been hugely helpful because it is longitudinal over a number of years. The multiple myeloma data is longitudinal. So it does allow us to now start to simulate trajectories of, at the individual patient level, and response to intervention at the individual patient level along a trajectory. And that's been super important, especially in diseases of time, so degenerative disorders like Alzheimer's and other neuro diseases.
Could we just be, I want to be provocative for a minute, so if you just, for a change, could you just go back 2 or 3 slides to your experimental slides, which you have to do now you've found the gene and you want to move forward? Because I want to make a plea for the experimentalist. Here, that one.
Here we go.
Right, so beautiful. I mean, I love the work's great. The way you can predict things and the way you can do the changes on the computer and get the answer that you can then check out. But the checking out is the interesting piece when you're thinking about how long it takes to do things. So these experiments, which a lot of people in the room have done, take a long time. They're biologically variable. So I think it's important that we put AI into the perspective of it, which it's very powerful. It gives you great starting points, but you still have to do experiments.
Yes, you have to do experiments, and you have to end up doing experiments on non-humans for obvious ethical reasons. And so I spent so much time at the beginning saying why it's so important that we're getting the data from human samples. And we're religious about starting with human samples at our company. So it does feel not so great to then have to go and do these experiments back in cell lines and stem cells and animal models, but you need to produce some level of wet lab evidence before you can bet $50 million on a clinical trial. And so we do take these experiments with a real grain of salt. They take a long time-ish. Some are a couple months, some are half a year. And they're expensive.
Yes.
Relative to now what's becoming very cheap and fast experiments you can do in humans. So I do look forward eventually to a time where we can do less and less of this. Look, it's important to check a box and do some of this, but if we really don't believe that some of these preclinical models, these cell line systems, are representing a complex human neurodegenerative disease, you could start to say, well, why do we do them? And why do venture capitalists, why does the FDA still require this check the box, where if it actually reads out well on some of these preclinical assays, it's obviously no guarantee it's going to work in humans. So I do look forward to a time where we're building a large enough number of these digital twins in a given disease from a large enough patient cohort such that you get to the point of cross-validating discoveries amongst 5, 6, 7 different independent patient populations from around the world. Does that now become enough of a compelling set of validations such that you don't really care what happens here? Because the question is, if it doesn't read out the way we want, do we kill the program or not? When we have the evidence from a much more relevant system and there's much other cooperating evidence. So the FDA just a year ago, I don't know if people paid attention to this, for the first time they allowed drugs to be entered into human clinical trials that have not gone through animal testing. So it's a big step. We'll see how quickly people adopt it, but there was a drug for postpartum depression that did just get approved that never went through animal testing.
That's very cool. I think that's a great place to stop. Let's ask further questions at the break, which is now. You need to be back by 11:20, and look forward to the conversations over a cup of coffee. Thanks, presenters, and